So the next speaker is Paul Grossfeld, is going to give a sort of a quick overview of heart problems. Va a hacer una revisión breve sobre los problemas cardiológicos de síndrome Jacobsen.
Gracias. One thing I want to say about Dr. Dahlm's talk, and that is these treatments can literally be life-saving, and we're already seeing some evidence that some children that are being treated are doing much, much better. So this is very, very important. Antes empezando, yo quiero decir gracias a Mónica para traduciendo. Muy importante. Okay. So I'm going to spend a few minutes talking about my area of relative expertise, and that's on the issues of the heart. So my interest really started back in 1995, when in my training I saw my first patient, a young child with Jacobsen syndrome. And in general, we know that only about 10 or 20% of children or people with heart defects have a known specific genetic cause. For example, like Down syndrome. So after I saw that first patient with Jacobsen syndrome, who I was taking care of because of his heart defects, I soon met Dr. Mattina and just a few others who helped to provide me with information about the types of heart problems these children have. And overall, I found about half of your children have some kind of a major heart problem. So I'm just curious, how many of you have a child that has a heart problem? Raise your hand. One hand per family. Let's see. Again, I just want to count. 1, 2, 3, 4, 5, 6, 7 out of 13 or 14. So that's right on. So of those that have a heart defect, we divide them into two classes. I have one class are those that have defects affecting the left side of the heart, or sometimes a hole in the heart that we call a ventricular septal defect. And you might remember I showed that slide in my first talk of those 4 children that have the most severe heart defect called hypoplastic left heart syndrome. And somewhere around 5 to 10% of children with Jacobsen syndrome are born with hypoplastic left heart syndrome. But very sadly, many of those children die shortly after birth. So the percentage of those surviving is less. Any of your children born with hypoplastic left heart syndrome? That's not surprising. In the United States, there are about 1,000 to 2,000 children every year with hypoplastic left heart syndrome, and about 1 or 2% of them have Jacobsen syndrome. And then this is a long list of many of the other defects that occur less commonly. So just to summarize, about half of your children are born with a major heart defect. There is a very high frequency of children born with hypoplastic left heart syndrome compared to the general population. And many of the most common heart defects that occur in the general population also occur in Jacobsen syndrome. So for me, this is very important from the research aspect, because we think that what we learn from children with Jacobsen syndrome will apply to many of the 1% of all children that are born with a heart defect. Make sense so far? So one thing that's very important that Dr. Mattina mentioned a little bit, for Jacobsen syndrome, what do we recommend? So any child with the new diagnosis of Jacobsen syndrome needs to have an echocardiogram, which actually shows the structure and function of the heart. And nowadays, Jacobsen syndrome can be diagnosed very early in pregnancy. And we can do a diagnostic echocardiogram of the heart of a fetus about halfway through pregnancy. Got that? How many weeks? If your echocardiogram is normal in the beginning, that's very reassuring, of course. But for some of these heart defects, they can develop over time. So it's very important to know that. So hopefully, if your pediatrician listens to your child's heart, some don't, if they hear a new murmur, soplo, murmur, then they should be referred again for another cardiology evaluation. That's very important. So again, some of these heart defects develop over time. So those are my clinical recommendations at this time. And if this is okay, what I want to do is tell you a little bit about our research. And as Dr. Martina mentioned in her talk, I'm going to give you an example of how understanding the differences between why some of your children have a heart defect and why some don't might help us maybe to find a way to prevent some of these heart defects in the future. And that can only come after many, many years of research. So like I showed you before, we have found a very small region in chromosome 11 that is missing or deleted in every child with Jacobsen syndrome and a heart defect. And as you see in the blue, that's a child with a deletion to the end of the chromosome, right there. And then these other ones are children that have many features of Jacobsen syndrome, but have interstitial deletions. And that helped us to find a very small region of overlap that only contains 5 genes. And I'm going to spend the rest of my talk telling you about the gene now that causes the heart defects in your children. And forgive me because I know this is a lot of information, but this is what we know and what we're learning about this gene. What we already knew about this gene is it's actually critical for the cells that Virgil talked about for the immune system. But nobody, until our studies, thought this gene might have anything to do with heart development. But interestingly, this gene is what's called a transcription factor. And transcription factors are proteins that are essential for regulating a number of very important biological processes, including the development of organs. And in fact, for the few genes that are known to cause heart defects, most of them actually encode transcription factors. So again, we turn to the mouse, our friend, and these are pictures of a heart from, this is a mouse fetus. This whole heart is only about 2 millimeters in size. And if you look at a normal heart, you can see all the normal structures. For example, the 2 main pumping chambers of the heart. And you see the main artery giving blood to the body called the aorta. And this is the artery that gives blood to the lungs called the pulmonary artery. And we see in one strain of mice that when we get rid of the gene, all of these mice have a hole in their heart in the wall that separates the 2 main pumping chambers. And we call that, again, a VSD, which means ventricular septal defect. And we also see that the aorta now is pushed over and lines up with the right side of the heart instead of the left side of the heart in the normal. So we're trying to understand how this happens. This is about the most boring slide that I have to show you today, but maybe the most important. Because what it shows is that whether or not you get a heart defect and what kind of heart defect it is depends entirely on which strain of mouse we study. Does that make sense? And different strains mean they're what we call different genetic backgrounds. So what this suggests is that there are other genetic factors that determine whether or not you get a heart defect and what kind of heart defect you get in the absence of this gene in 11q. And for you, this we think will help us to understand why Elliot was born with a very complex heart defect that required open heart surgery when he was just 1 week old, and why Amy, who has the same deletion as Elliot, was born with a perfectly normal heart. So now I want to tell you, if I can, a little more about what we think this gene is doing to cause these heart defects. I'm getting the warning. It turns out that when the heart is developing in the womb, there is a very special population of cells called the cardiac neural crust that starts in the developing nervous system of the embryo. And these cells move out over time to form different structures of the heart. And what we found is that in the absence of the ETS1 gene, those cells aren't able to move out to the heart normally. And we have a way to study that in a petri dish. And what's shown here is some of that tissue from a normal fetus of a mouse. And each one of these dots shows those cells moving out over time. And what happens, though, in the mouse that gets a heart defect, in the absence of the gene, you see far fewer cells, and they're not able to move out as far. And this is a very busy slide, don't worry about it. But these are actually looking at those same cells in an actual embryonic heart. All those cells are stained in blue. And what you see, let's say, as shown here in the normal heart, a lot of blue on both the left side of the heart, the aorta, and the right side of the heart, the pulmonary artery. And what we see when you lose the gene is that there are far fewer cells overall making it out to the heart. And it seems to be affecting the structures on the left side of the heart. So just to summarize, the loss of this gene causes a decrease in the number of these cardiac neural crest cells that leave that neural tube. They are not able to move as well. And the overall effect is that there are fewer of these cells going to the heart. And we have some evidence suggesting that they might instead go to some other places, like to the head. Oh, I'm going to skip this just to show you that. I'm just going to show you that we can actually follow those cells here in green, moving in real time to the developing heart in an embryo. And this is just to show the beating heart of what I just showed you in a developing heart from the embryo. So I just want to say thank you to 3 very important people in my lab that have done these studies. And I will be happy to take questions. And I must say, this is my most important human genetics project.
Thank you, Paul. I appreciate that you're all sort of talking at a slower pace, you know.
I speak too fast, I know.
No. I'm saying no. I appreciate,
Yeah.
you know, that you're actually talking slower. So that's, thank you. Okay, so, Teresa?
Any questions first?
Do you want to talk about hematology, or maybe leave it for the discussion, for the open discussion?
Yes, for the discussion.
Okay, sí, entonces la profesora Martina, la cuestión, los aspectos hematológicos los comentamos luego, en principio en la... Okay.